Hi, I'm Shoji Tsuji, presenting from Tokyo, Japan. I would like to show recent progresses regarding the clinical trial of Ubiquinol for multiple system atrophy. Here is my disclosure. MSA is a sporadic neurodegenerative disorder with late adulthood onset. Clinical presentations are characterized by various combinations of autonomic failure, several symptoms, Parkinsonism, and pyramidal signs. Two subtypes, MSA-C, presenting with predominant cerebral ataxia, and MSA-P, presenting with predominant Parkinsonism, have been established. Accumulation of alpha-synuclein has been identified as the pathological hallmark for MSA, but the etiologies of MSA remain to be elucidated. MSA has been regarded as a sporadic disease without any familial occurrence. Before going to the clinical trials, let me briefly outline the background of genetics of MSA. As I mentioned, MSA has been regarded as a sporadic disease without any familial occurrence. Although the frequency is extremely rare, we have identified multiplex MSA families. Furthermore, we have identified MSA patients with Parkinson's diseases in the siblings or parents of the MSA patient. These observations strongly support that genetic basis underlying MSA. So far, we have identified 6 multiplex MSA families as shown in this slide. And in pedigrees shown in this slide, affected individuals are all sibling cases. Parental consanguineous marriage was observed in one of the 6 MSA families. Furthermore, we have experienced 11 multiplex families where the MSA patients have siblings or parents with Parkinson's disease, as shown in this slide. Thus, MSA and Parkinson's disease may share common genetic factors. This slide summarizes the recent studies showing decreased coenzyme Q10 levels in MSA. In multiplex MSA families, we have discovered biallelic CoQ2 mutations in multiplex families. In sporadic MSA cases, we demonstrated that CoQ2 V393A confers a risk for sporadic MSA cases. Furthermore, decreased coenzyme Q10 levels have been demonstrated in plasma, cerebrospinal fluid, and cerebellum, even in patients not carrying CoQ2 variants. So taken together, these findings indicate that the decreased coenzyme Q10 levels commonly underlie MSA not only in MSA cases with the CoQ2 variants, but also in those without CoQ2 variants. And of the 6 multiplex MSA families, we discovered biallelic CoQ2 mutations in 2 families, in this family and in this family. CoQ2 encodes an enzyme involved in the biosynthesis of coenzyme Q10, and the mutations in CoQ2 gene are expected to result in decreased synthesis of coenzyme Q10. And indeed, we observed dramatic decrease in CoQ10 content in the cerebellum of a patient with biallelic CoQ2 mutations. We then investigated if CoQ2 gene variants are associated with increased risk for MSA. And we identified that carrier frequency of CoQ2 B393A variant is 9.1% in MSA cases in Japan, but the carrier frequency is only 3.3% in Japanese controls, giving the odd ratio of 3. When we published the association of CoQ2 B393A with MSA, it has been quite controversial because similar associations were not observed in MSA patients with European descent. However, association of CoQ2 B393A with MSA has been reported mainly from East Asian regions, including China, Taiwan, and Japan. This slide shows the results of a meta-analysis of association studies of CoQ2 B393A reported in East Asian regions. And this meta-analysis confirmed a strong association of B393A with MSA in East Asian regions, including China, Taiwan, and Japan. Given the strong association of CoQ2 B393A with MSA in East Asian populations, we then investigated the distribution of CoQ2 B393A variants in various populations, and as expected, we found a high carrier frequency in the Japanese and East Asian populations with an allele frequency of 1.5%. In contrast, the allele frequency of this variant in the populations with European descent is extremely low or virtually absent. And intriguingly, we found that the carrier frequency of this variant is much higher than East Asian populations in Latino and mixed American populations. Therefore, it would be interesting to see if the association of B393A with MSA is observed in the Latino and mixed American population. After we published CoQ2 variants in multiplex MSA families and association of CoQ2 B393A with MSA, decreased coenzyme Q10 levels in plasma, cerebrospinal fluid, and cerebellum have been demonstrated even in MSA patients not carrying CoQ2 variants worldwide. Thus, decreased coenzyme Q10 levels seem to be a common finding in MSA. The reason why coenzyme Q10 levels are decreased in MSA patients not carrying CoQ2 variants, however, still remains to be investigated. As summarized in the slide, decreased coenzyme Q10 levels commonly underlie MSA, not only in those with CoQ2 variants, but also in those without CoQ2 variants. These findings suggest that supplementation of coenzyme Q10 may serve as a therapeutic approach for MSA. This slide shows the function of coenzyme Q10 in mitochondria, shown to the right. Coenzyme Q10 serves as an electron transporter, and decreased coenzyme Q10 is considered to result in decreased production of ATP in mitochondria. In addition, coenzyme Q10 has potent antioxidant capability and served to protect neuronal cells against oxidative stress. Thus, supplementation of coenzyme Q10 is expected to serve as a disease-modifying therapy for MSA. From these 2 points. To explore the possibility of coenzyme Q10 as a disease-modifying therapy for MSA, we first considered a high dose of coenzyme Q10 would be desirable. There are 2 redox states of coenzyme Q10, reduced ubiquinol and oxidized ubiquinol. Since bioavailability of ubiquinol has been shown to be higher than ubiquinol, we considered ubiquinol would be a good candidate. Through consultations with PMDA, a governmental agency for approval of new drugs in Japan, we were asked to conduct a phase 1 clinical trial to confirm the safety and pharmacokinetics of high doses of ubiquinol. With this background, we conducted phase 1 trial of ubiquinol starting in 2015. We first conducted phase 1 clinical trial of ubiquinol to confirm the safety and pharmacokinetics of high dose of coenzyme Q10. This slide shows the outline of the phase 1 trial. Healthy adults, male volunteers, were given 900, 1200, and 1500 milligrams of ubiquinol for 14 days. In this phase 1 trial, we did not observe any serious adverse events, confirming the safety of high doses of ubiquinol. As shown in the panel to the left, substantial increase in plasma levels of coenzyme Q10 was observed. Furthermore, the substantial increase in ubiquinol in the cerebrospinal fluid were also observed, shown in this slide. Having confirmed the safety of high doses of ubiquinol, we then proceeded to the phase 2 trial of ubiquinol for MSA. To smoothly recruit MSA patients into the clinical trial, we first established Japan MSA Registry. The aim of this registry is to obtain the detailed natural history of MSA and collect bioresources to establish a biobank of MSA. In this registry, clinical information including the UMSA-RS Part 1 is obtained every 6 months by telephone interview, and patients are asked to visit institutions annually to receive examinations by skilled neurologists, including the UMSA-RS Part 2, and bioresources including plasma and dynamic DNA are collected and deposited to Rare Disease Bank operated by National Institute of Biomedical Innovation. All the clinical data are stored in the cloud database and made available to the future clinical trials. In this registry, 529 MSA patients have been registered so far, and longitudinal clinical information is being collected as I presented in the previous slide. This slide describes the study design of multicenter phase 2 clinical trial of Ubiquinol for MSA. 13 institutions in Japan participated in this study, and MSA patients who fulfilled probable or possible criteria and could independently walk 10 meters were enrolled. MSA patients were stratified based on genotypes of CoQ2, including 20 patients with B393A and 100 patients without B393A. And MSA patients were randomly assigned to MSA or 01, Ubiquinol, or placebo with a ratio of 1:1. The primary efficacy outcome was the changes in the UMSARS Part 2 score from baseline to week 48. And secondary efficacy outcomes include UMSA Part 1 Basel Index, scale for assessment and rating of ataxia, SARAS scores, and 10-meter walk time from baseline to week 48. As shown in this map, 13 institutions from all over Japan participated in this clinical trial. This is the study flowchart. Starting with 300 milligrams per day of Ubiquinol, the dose was increased every 2 weeks up to 1500 milligrams, and this dose was maintained for the remaining 40 weeks. After randomization, 63 patients received Ubiquinol and 68 patients received placebo. Due to the pandemic of COVID-19, we experienced various difficulties in conducting the clinical trial, but despite these difficulties, last patient out was accomplished in 2020. And we are currently analyzing the data in detail and hope to publish the results very soon. Finally, I'd like to thank all the MSA patients participated in this clinical trial and many collaborators as shown in this slide. In particular, I'd like to thank Dr. Jun Mitsui, who played a central role in this clinical trial. Thank you for your kind attention.